joining us online tonight. Thank you for joining us, and we're sorry for the technical delays, and also for our wonderful live audience here tonight. We're apologies for keeping you waiting. But I'm just going to kick off with a karakia, and this is about seeking knowledge. So, e te hui, whaia te mataronga kia marama, kia whai take nai mahi katoa, tu maia, tu kaha, arahau atu, araha mai, tātou I a tātou katoa. And this means, for this gathering, seek knowledge for understanding, have purpose in all that you do, stand tall, be strong, let us show respect for each other, which is very fitting for this type of speaker series. So a very warm welcome to you all, if you're here in person or online, and this is the third in our 70th anniversary year speaker series, and we are very privileged to have Dr. Amy Chan, or Associate Professor Amy Chan, and Dr. Tim Angeli-Gordon with us tonight, who will be presenting on respiratory and AI and asthma, and also gastrointestinal disorders and diagnosis. So, but we have Dr. Julie Lim and Associate Professor Vanessa Selak to do the introductions, so I won't introduce our guests right now. But look, thank you for coming along tonight, and as mentioned, we have the 70th year celebrations this year, and we are celebrating over $100 million worth of funding towards medical research from Auckland Medical Research Foundation. And for those of you that are donors in the audience, you were the ones that helped make this possible. So a huge thank you from us, a huge thank you from our research community. You have made possible life-changing differences, so big thank you to you. I'd also like to thank our partners that have joined us for our 70th year celebrations, which are the University of Auckland, and we've got Dr. Frank Bloomfield here tonight, and Nina Bowen, also from the University of Auckland. So thank you very much for being here tonight and for your partnership. Auckland University of Technology are also a partner, along with JMI Wealth. And we couldn't be doing what we're doing this year for the celebrations without them also. So thank you very much. So for those of you that don't know me, my name is Sue Brewster, and I'm the Executive Director of Auckland Medical Research Foundation. Lots of people have heard me say that there's 5 of us in the team, and we're all executive directors, because that's how good the rest of the team is. And they're all here tonight, and they make the wheels go round on this wagon. So thank you, team, as well. So I know why you're all here, and I am not going to take up any more of your time. We'll have Amy presenting first for approximately 40 minutes, and then we'll do questions and answers, and then we'll be followed by Tim with more question and answers. But please, housekeeping, bathrooms are up the stairs, either way you'll find it, and emergency evacuation if you go up the stairs in an orderly fashion and meet out the front. You'll find me there, because I would have dashed up before you. So look, thank you so much, and I'm going to ask Dr. Julie Lim to come forward, please, and do our first introductions for the night.
Thank you, Sue. Well, good evening, everyone. My name's Julie Lim, and I'm a member of the AMRF Medical Committee. And it's my pleasure tonight to introduce our first speaker, Associate Professor Amy Chan. So Amy is the respiratory lead of the Medicines Intelligence Group at the School of Pharmacy, and is soon to take up the role of Head of School. She is a clinical pharmacist by training, with over 15 years of experience in hospital practice. Her research journey began with a desire to better understand asthma, leading to a PhD focused on the use of digital technology in asthma management. Her work at University College London further demonstrated how digital tools can reduce the risk of asthma attacks and enhance the quality of life for those living with asthma. Amy is currently the recipient of the AMRF Senior Research Fellowship and leads a dynamic research group that sits at the intersection of digital technology, big data, and respiratory health. We are delighted to have her with us this evening and very much look forward to hearing about her latest research developments. Please join me in welcoming Amy.
you, Julie, for that very kind introduction. Bring up our slides. Right, so first of all, I really want to thank you, everybody, for making a time this evening to come and listen to my story and our story. And very much I wanted to share as a story that has very much centred around Auckland Medical Research Foundation. We know it's world-class health research. They've talked about how they've empowered researchers like myself and really transformed lives. So I'm going to start with a video that will share a life of a patient that has been influenced by the research that we are doing.
I was having asthma problems. I thought I had it all together, and I didn't. When you're in the corner and you're trying to sit down and you're basically just keeping yourself alive. You're like, all I care about is that as long as I can take my next breath. That's the scariest thing, but you don't know how to communicate that with the people that are sitting in front of you. So it hasn't been a great journey. It's been very tough, and I've pushed myself to the next level where I shouldn't even be standing.
And some of you may recognise that this patient who suffers from asthma is the famous celebrity boxer Mia Motu, and she shared with us her heartbreaking journey and her story and her struggles against asthma. And it's with that backdrop that I have the privilege and honour of bringing you today the story of how we could harness the technologies we have to help people with asthma, such as Mia. So tonight in the story, I'm going to structure it easy, 5, 4, 3, 2, 1. Hopefully there's some facts that we can take away. But I'll talk a little bit about 5 reasons why we think asthma matters, 4 facts about asthma attacks that have really informed my journey, 3 discoveries around biomarkers, particularly around the concept of digital markers, 2 key types of digital markers that my particular research has focused on, leading to the DigiPredict study that is funded by Auckland Medical Research Foundation. So I'm going to convince you today and then why asthma matters, and it's not just about breathing. So we know that asthma is affecting somebody somewhere right now, and the rates in the literature vary somewhere between 260 to 330 million people globally that have asthma. And is very much a leading cause of school and work absences, primarily because it is predominantly a disease of childhood, but also we see very severe asthmatics all the way up into adulthood. And actually it causes approximately 450,000 deaths a year, and that is a number that has not really changed since 2006, so our death rate or mortality rate hasn't improved despite available treatment. So back home, if we look at the numbers, if we took the population of New Zealand and just the population with asthma, we know from our research led by our own group that there is an attack happening every 2.5 minutes. There is a death from asthma every 5 days, and this is often underestimated and not talked about as people don't realise the magnitude of asthma and its impact on our population. The cost to society is over $1.2 billion from both direct and indirect costs, and about 14% of our population continue to come through as frequent flyers through our hospital system, being readmitted every month with really uncontrolled asthma. And it doesn't take much. These are just newspaper clippings that I took recently. And you can see that asthma is still very much hitting the headlines in terms of our youth in particular who are dying from unpredictable and sudden asthma attacks. In our research of concern, we also have shown that in the last 10 years or so, even though the prevalence of asthma is relatively stable, about 1 in 8 of our population have asthma, the rates of control have become worse. So the rates of asthma attacks have increased by 1/3 in the last 10 years, and we're not seeing that improving despite available treatments. And really concerningly, asthma attacks are a key source of inequity. If you can see the rates across Māori Pacifica, they're much higher, and the increases are very high across the 10-year period in Māori. You can see here from this particular figure, the highest rates are in Pacifica, followed by Māori, and there is a clear socioeconomic deprivation gradient as well, where the most and highest rates of asthma attacks occur in the most deprived patients. But the greatest increases, however, have been in the least deprived patients. So we know across the board, asthma attacks do affect everybody. In recent work as well, we've also shown there is geographical region in attacks across the motu, across the country. So the red are the hotspots of asthma attacks, which we measured based on oral steroid dispensing data. And the hotter or more red the spots, the greater the number of asthma attacks in that region. And you can see there is really a rural-urban disparity with greater disparity in rural areas, and also a significant distribution effect as well across the North and South Island. But perhaps most importantly, why asthma attacks matter are the patient experiences that we hear. And this is data from interviews that we have done, as well as interviews that other research groups have done. And you can see that having an asthma attack is really, really of concern to a lot of patients. They talk about the fact, they thought they were going to die, and that kids sometimes sit there watching their mum, struggle with breathing, and it really affects, obviously, the experience of both the children and the mother. They talked about airways being blocked by a feeling of like a cement brick. It was really hard, really tight, really gasping for that next breath. And they talked about the effects on sports and hobbies, that they couldn't had to give up their sports and hobbies as a result of their asthma, that it got so bad that they couldn't even walk. And the coughing in particular drains people out. They talk about the coughing really depleting the energy, lasting sometimes for days, up to 10 days. And that a lot of people in particular think that asthma is not taken seriously, which makes it really tough for people with asthma, because people don't know what to look for, and also they don't believe that they have a serious condition until they're literally on the floor. So hopefully I've convinced you that asthma is a really important problem to talk about. And the next part that I want to discuss is around the facts about asthma attacks. So in terms of looking at the history of asthma, so where does asthma come from? It actually comes from, in the Greek times, from the word asian, meaning panting or shortness of breath. And you can see evolution over time from a description of the core asthma symptoms around panting. They also discovered triggers such as pollen back in 1870 as a cause of what they called hay asthma, which has kind of evolved to hay fever. And then in the 1920s to the present, the evolution that has been established, the links between asthma, inflammation, and modern treatments. And over that time, what is core, despite the revolution in treatments, core to this is still that the symptomology, the panting, the shortness of breath, the difficulty in breathing, that has not changed despite the advances in treatment. And also the suddenness of the asthma attacks, that still remains since Hippocrates times. So asthma attacks, we know, is related to things like emergency department visits and hospitalizations, is linked to more days off school and work. But most importantly, asthma attacks are a leading cause of preventable deaths in New Zealand and worldwide. And perhaps one of the most interesting and important facts is that asthma exacerbations are not only an important goal and target for treatment, but it's also associated with progression of asthma. So those people who have asthma attacks, every attack that they have increases the risk of them having the next attack and the following attack, which is why it makes sense the people that we are seeing in our hospital system and in our clinics are often the frequent flyers, because every attack that they have could potentially be leading to more damage to their lungs and a rapid decline in lung function. The good news, though, is that if we look at the timeline of an attack, is that it doesn't happen as suddenly as we think. There is often a trigger that happens, and then the peak attack or exacerbation event, which is where most people focus their energies on, and that's when they present to hospital. But actually, there is this variable onset phase, which can last anything from minutes to days to potentially weeks, We don't know what that speed of onset is, but we know it is variable, and there is a chance perhaps to intervene in this onset phase to abort the entire process of an exacerbation. And perhaps this is where we can save lives by helping signal when a person has an increased risk of an asthma attack. And we also noticed from a very important piece of work done by our colleagues in the UK, which is the UK National Review of Asthma Deaths. So what they found is over 95% of asthma deaths actually occur in adults with known risk factors. So these are people who have known risk factors for attacks. And 85% of deaths don't actually occur suddenly, as we think, as is depicted often in movies, but is actually progressively worsening over days and weeks. So this is one of the key recommendations on their report, which we wanted to act on. The key advice was know your risk of having an asthma attack. And I was like, this is easier said than done. And we could tell our patients, you just need to know when your risk is worsening. But actually, if you look at the evidence, at least half of the people who died did not know about their risk, and they were just being treated for mild forms of asthma or moderate asthma. It's not the severe forms that come to hospital that's under specialist care that we're worried about. It's the mild to moderate people with asthma that may only have symptoms 0 to 1 times a month that are dying from asthma. And in particular, this particular statistic hit home for me from the report, that poor recognition of risk of adverse effects was found to be an important modifiable factor in 70% of children and over 80% of young people. They simply just didn't recognize how severe their asthma attack was and how important it was to seek help and intervene. And I think we can all know people how busy lives can get. It's just so easy to adjust. You've got a bit of breathlessness, you want to keep going. Life is busy, you don't want to stop to make a medical appointment, or you might have to wait days before you get to see your GP. And actually that time is lost, and this is where we could intervene to signal earlier to save the lives of these preventable deaths. So this is where I started my journey to action that particular recommendation. What can we do to help identify people at risk using digital biomarkers? So if we take a little journey down into people's airways, so we know asthma is a combination of issues with breathing. There's in the lungs, there's inflammation, and down in the airways. And what we can see is there's increasing mucus and airway inflammation, and airway is tightening over time as the risk of asthma attack increases. But unfortunately, unbeknownst to a lot of people with asthma, you have to reach a particular threshold before we recognize the symptoms are bad enough to act upon. So what we are essentially doing when that happens is we're waiting for the fire alarm. So, as inflammation builds and the symptoms appear, we only react when the symptoms reach a particular threshold, and all the current guidelines and the asthma action plans that we have are symptom guided, and we only act to increase treatment or seek help or see a health professional when symptoms get worse and an attack occurs. But really, the key point is, what if we could see the smoke before the fire happens, very much similar to a fire alarm? And this is where the analogy happens with the work that we've been doing with digital markers. As the inflammation builds, as I saw in the airway, and it's often silent, we could get data insights now that can provide insights into what's happening in the body based on these digital biomarkers. And as the symptoms start to appear, we can actually take preventative action already before they reach the point that the symptoms become so bad that they reach an attack point. And ultimately, we hope that will help change care from reactive to proactive care. So, in summary, the opportunity we're looking at is that because attack risk increases over days to weeks, there's this untapped opportunity to intervene. And what AMRF has allowed me to do is really focus on this point to see, are there digital markers I can detect using everyday technologies, and can we develop a risk alert system using these everyday technologies? So what can we do to sense? Basically, we know we have traditional biomarkers, which I'm sure most of you are familiar with. So things like taking a blood test, a tissue sample, or taking a peak flow or exhaled nitric oxide measurement as a marker of inflammation in the lungs. But the problem with these traditional biomarkers is they're not often accessible. And if you think of the analogy, it's a bit like you can take a photo, which is only a cross-section in time, but we don't know what's happening over time in between those photos. What digital biomarkers allow us is an ongoing continuous video feed. So no longer do we only have snapshots in time of what's happening to people. We can actually have a live video feed to actually understand what the person's risk is and physiologically what is happening. And this is quite interesting. This is just an example from an Apple Watch of a cardiogram. But you can see they look so varied, and there's so much data in each of these cardiograms, like the way someone's sleep looks without alcohol, and then with dinner, alcohol after. Sleep is very much affected, and the patterns look different in someone who's had alcohol. Likewise, we can say there's fitness data, but fitness data looks different in people who've done regular running episodes as opposed to someone playing continuous sports games such as hockey. And same, all of you who came here in rush hour to come and see us, the stress of driving in rush hour compared to going for an interview, for example, the stress is more sustained during a drive in rush hour compared to if you were going just for an interview. And with illness, again, atrial fibrillation looks quite different to atrial flutter. And are these learnings that we're trying to draw on to help develop this particular digital prediction alert for asthma attacks. So talking a little bit about digital markers. So digital biomarkers have been recognized by the FDA formally since around 2022. And the officials have recognized that often people have misused the term digital biomarkers. And now they've actually set on a set definition of what a digital biomarker is. So when we look back at the basics of detecting disease, often we start with patient questionnaires or symptomology, so that's often outcome assessment. So those are COAs, where we focus on consultations or outcome assessments to assess how patients feel using outcomes, using questionnaires. And there are true biomarkers, like the traditional biomarkers I've talked about, so visiting a lab, taking a blood test. And then what we're trying to focus is that in between using a digital health technology, DHT-derived measure. So how can you use technology to capture some of those physiological changes and some of those behavioral changes? So instead of just using questionnaires or taking blood tests or labs, can we use this technology to develop that is developed around us to detect both behavioral and traditional changes? So digital biomarkers, the formal definition from FDA is basically a characteristic or set of characteristics that's been collected from technologies, and it's used as an indicator of changes or responses to an exposure of intervention or therapeutic interventions. And this is where digital biomarkers really come in from consumer-grade devices. They allow us to collect insights 24/7 into what's happening into people's bodies, so we can provide real-time alerts, forecasts, and personalized results specific for that particular individual. And actually already exists for a number of different conditions. So FDA, I think, approved the first digital marker in clinical trials around 2022 for Parkinson's disease. So it's been used to look at motor activity and looking at tremor based on a wristwatch. It's also been used for things like neurological conditions and metabolic diseases. It's been used in COVID and sleep disorders. But really, what about respiratory or asthma disease? And so this is where we're trying to see, what is the use of digital markers when it comes to helping, share decision-making, helping to triage patients, for example, helping to monitor the therapies that they're on, and also thinking about when to start or stop treatments, particularly some of the more expensive treatments, such as biologics. When should we be starting these and when should we be stopping them? So Julie, in her kind introduction, mentioned about some of the work I had done at University College London. And this is one of the large Cochrane reviews, many hours of looking through the literature. But we really found that digital technology can play a role in helping manage asthma and potentially halving the risk of asthma attacks. And it is from this Cochrane review that we then moved into this phase of research to see how can we actually harness that particular potential. to really help predict asthma attacks. So meet Jay here. He's actually in the audience, one of our team members. But let's take an example. Jay here has particular love of technology, and he's got a range of different types of smart technologies. And this is to show you that these technologies exist. There are smart glasses that already exist that can track eye movements. There's smart shirts, and take samples of your sweat and do a physiological evaluation. There's smartwatches, which I'm more sure you're all familiar with. There's also smart shoes that look at your cadence, your walking patterns. That's been used in Parkinson's disease. Smart socks also to look at your sweat patterns, your volume. Do electrolyte assessment if you wish for that to happen. There's also things like smart belts, baby controls. I know they've used it to see if they can prevent cot death. There's little anklets that you can give the babies to look at how much movement they have in the cot, and to sense things like respiratory rate, and also things like smart rings as well that also look at the same parameters as smart watches. So these all do exist. They're not something in sci-fi movies in 2050. They already very much exist nowadays. And so what can these smart technologies collect? Well, physiological parameters, behavioral parameters as well, so it can monitor things like medication use, activity levels, sleep patterns, environmental as well. So by tracking location, we can link directly to environmental data, pollution, air quality, and these are all very important data points when it comes to respiratory disease. And just to contextualize device ownership rates, so year on year, about a third of our population already owns smartwatches. I'm sure many of you in the audience may already have a smartwatch that you're currently wearing, and definitely smartphones. And year on year is one of the greatest and most massive changes year on year. It's growing in its market size about 10% every year. And there's New Zealand, which is roughly the worldwide average. It's roughly ranging, I think the updated stats are more close to about 30% in terms of people who own a smartwatch. So now onto the actual juice. Now, what are these digital markers that we're looking at? So this is the study that we've been doing with AMRF support. So what we've been doing is we've been giving people with asthma in New Zealand, and we've been asking them to monitor their peak flow, to use a smart inhaler, which will track their inhalation patterns and their medication use. They all got free smartwatches, so they were very happy to take part in the study. And they also got a smartphone app to really monitor their symptoms over time. And what we were hoping to do was link it with data from the Ministry of Health and environmental data sets from LAWA/NIWA, Stats New Zealand, to generate this clinical risk score, which will give you an asthma status at a glance and provide insights into a person's asthma attack risk. So what we realized, really it comes down to two types of digital markers. It's either symptom-based or physiological-based markers. So symptom-based markers are things that are driven by symptoms. So we know that cough and wheeze, as coughing goes up, people's inhaler use naturally also goes up, and we can now capture that early on using smart inhaler technology. And you can see as it goes up, the peak flow also goes down. So that's a physiological marker. The other important symptom-based marker is in the last probably around 5 years or so, there's been cough monitoring apps, AI cough monitoring apps that exist. You can look them up on the App Store. There's a couple of different competitors out there, but they mainly use the inbuilt microphone in your phone, and it's trained only to detect cough, which we tell patients it doesn't detect conversations, but it is trained to only detect cough sounds. It can count the number of coughs and also give you an idea of whether they're dry or wet coughs. And in particular for asthma, we were particularly interested in nighttime cough, because we know nighttime cough is a cardinal symptom of worsening asthma. And what we did see here, in the left-hand side, is the cough count over time relative to the date of the attack. So 0 was the date of the attack, and you can see there is an increase in the median number of cough counts leading up to the attack. Or when we looked at a mean cough count, this is what we saw in patients, is that there is an increase about a week, 7 or 8 days before the attack, leading to a peak potentially in the day before they reported the attack to us. And again, you can see here, we looked at the mean cough count over time, and -1 is where it peaks, and 0 was a day where they reported it to us. So we felt that this was quite an interesting marker of worsening asthma, potentially up to 7 to 10 days before the self-reported attack occurred. And again, this shows you basically the confidence interval, but across the sample of, I think there was 140 in this particular sample, you can see there is that pattern in cough leading up, in the days leading up to an attack. There's also rescue inhaler use that I mentioned, so you can see, and this is not my work, in America they gave people, again, a rescue smart inhaler, and they were able to monitor the number of doses of rescue inhaler use, and you can see it increased again around that 7-day mark leading up to the attack. So now you can see there's a bit of a story with that 7-day mark. And same with physiological measures. So here, this is actually not a digital study and done in the 1990s, but probably one of the most important studies that actually identified these key markers to show that actually before an attack happens, which is on day 0, there is a fall in peak flow and an increase in asthma score. So even back then in the 90s, they were recognizing that asthma attacks, there are markers that happen perhaps in the days leading up to the attack. And we were able to see the same here. So this is an our study sample in DigiPredict. You can see again about that week, there's that drop in the peak flow around the attack period and in a recovery period after. And we also got, using these smart inhaler technologies, we were able to capture things like inhalation parameters, which we weren't able to capture before. This is from a US study, but we saw very similar data that leading up to the attack, there was a drop in inspiratory flow, which was really interesting for us because asthma management has focused a lot on expiratory flow, so being able to blow as hard as you can through a peak flow meter, but it suggests that actually inhalation and inspiratory flow may also be impaired in the days leading up to an attack. And that's important because if we can put a sensor on people's puffers, they need to take their doses of medication anyway, so we can measure their inhalation as they take the medication. That could be an easily accessible digital marker as well of asthma attacks. It comes to heart rate. So here in the sample, you can see from an overseas study, they found in general that people with asthma had tended to have lower heart rates, and when it comes to post-exercise and heart rate recovery, which is what this shows, that they took longer and the change in heart rate recovery was smaller compared to the control group. And again, it's mirrored again in this other study, which looked at things like step count and sleep duration, and it looked at controlled and uncontrolled asthmatic and the healthy population, which is in red. And you can see across all parameters, step count in general is lower in people with asthma. Physical activity as well, and heart rate, you can see there's a change of heart rate. That heart rate seems to be higher in people with uncontrolled asthma compared to those who are controlled. And also sleep is also affected, and which is not surprising because of nocturnal cough in those who have controlled or uncontrolled asthma versus a healthy population. So we've been really focusing on these particular parameters, but again, day 0 is the day of the attack. You can see a reduction in step count, an increase in nighttime heart rate, and then a drop following an attack, and that all seems to coincide with drops in lung function and an increase in symptomology. Which brings us to the exciting DigiPredict solution. So I think where we're at is that what we want to do is use a smart watch and link to your phones to develop a risk score, and that risk score will continue to learn from your individual parameters, so that will be your personal risk score for your particular type of asthma. And this study will be the world first that's bringing together the biology, the physiology, and environmental factors. And we're trying to put this all together to look at how we can produce a personalized alert system. And unlike any other alert system, what you can see here, the attack risk is really personalized and is more accurate because we're drawing on multiple data sources. So this is an example of an example patient, day 0 being the date of the attack. You can see the heart rate variability might drop before the attack. So heart rate variability has been shown to correlate with inflammation. The lower your heart rate variability, the less healthy you are technically. So that's dropping before the attack. You can see sleep going down. You can see pollen going up potentially, the coughs going up, and the inhaler use also going up. So it's that picture that we're trying to capture with these digital markers. And I'm pleased to share, and this is hot off the press, this is data from our study so far, using the data we've collected so far from our patients. And we were really surprised as an investigator team at the accuracy and the performance of the smartwatch-only model and the poor performance of self-report. And this is really eye-opening because current clinical gold standard is we rely on things like the asthma control test and self-reported symptoms to predict attacks. And you can see it has really poor correlation in terms of being able to predict an attack. But surprisingly, using just the smartwatch parameters, you can see the RROC performance. The closer it is to 1, the better the performance. It's got really good performance just based on those parameters. We also did some really important engagement work with support from AMRF and CMDT and MedTech Consortium, and they found out through interviews with Samoan, Tongan, Pacifica families, Maori families, they said that they're really enthusiastic about the potential of this technology. They think that, you know, it's great that we can shut down the potential of an attack from happening. A 76-year-old kaumatua told us that they thought it would benefit mankind immensely, and that it was really important for whānau members, and they felt that the technology wouldn't be a barrier because they said it was something that could help Aija in our family. If their grandma was asthmatic, then everyone would help pitch in to help monitor the asthma and see how they could help their grandma. So this is a long journey. This is where we're at. It started, you know, in 2021 when we started using big data sets to really characterize the size of the problem for New Zealand. We then went out to communities to try and understand and explore the acceptability of this technology. Then that led to this key seminal study funded by AMRF, looking at identifying these novel digital markers of asthma exacerbations. It says 165, but actually this morning I think we're actually 173. And we think we'll get to the 200 because all 200 actually have booked in to see us by the end of August. So what we hope to do is build this prediction model and then bring it back to help people monitor asthma and provide alerts. So the next step to this is we're going to do a randomized controlled trial, which AMRF is also supporting, and then we hope to see how does this actually compare to standard care so we can actually implement it in routine care. And this is all just clippings like, you know, the smartwatch potential is great. It's been used already, especially for cardiac alert notifications. So potentially we can now expand it to respiratory and asthma. And I just want to say this has really been an enormous effort. The partnerships are worldwide. We've had support from so many people. And in particular, shout out to the team today here, some of them in the audience. We've had a lot of collaborators from clinical leaders, data science, Māori and Pacifica engagement, from primary care, such as National Hauora Coalition, Tend Health. And also really want to talk about my senior research fellowship. I looked back in it's year 3 of my fellowship, and in just 3 years, this is what we've really achieved. We've had so many students that have been supported. You know, we've had 9 research assistants that have been part of this project, 2 research fellows, 5 PhD projects as part of this. We've managed to get some more grant funding to help with grow and conduct this study. For example, we were able to make Fitbits free and smartwatches free for people in the study. And we've also had so many other outputs and a lot of media and international engagement. So really big thank you to AMRF. So as we know, hopefully I've managed to convince you that, you know, struggling to breathe is not okay. And if you'd like to scan the QR code here, thanks to Dr. Jay Gon in the audience. He's our AI and our marketing lead, but he's helped develop the prototype of what this app might look like to envisage it. So if you scan here, you can have a look at what we think that score might look like in the years to come, maybe next year when we go to trial. So yes, just really want to thank you all, and I'd love the kōrero after and the discussions. So thank you very much for all your support.
as fast as she talks, so and just as hard. So that was absolutely phenomenal, Amy, and everything that you've achieved in the last few years since we've supported you has just been incredible.
Thank you, Sue.
And Amy was recently promoted to head of department for pharmacology.
For pharmacy, yes.
For pharmacy. Yeah. So another accolade to Amy. So, but we have got time for questions because Amy went through all of that. So please, is there the audience? Might take me a bit.
Hi Amy, that was a great talk, thank you. And also kind of close to home because I know a few people in my family that have asthma. One question is, what is kind of the average age at which people are diagnosed with asthma? And is this something that could be used for younger children, or is it more kind of aimed at adults?
It's a great question. So the question was for the audience on the live stream. They wanted to know the average age of diagnosis for asthma and whether this technology could be used for people who were younger all the way to adulthood. So asthma, the incidence is actually, there are two peaks. It's often a childhood condition. So most asthma happens, around that kind of 5-year-old, younger kind of childhood period. But it does go all the way to around teenage years, and then often there is a dip or the growing out of asthma that you might have heard, but there is a second peak that happens around 40s, and often much more severe onset asthma or adult onset asthma that we often see. So it's technically bimodal, so there's two peaks. In terms of the technology, so the study we opened to 10-year-olds and above with no upper age limit. And so the oldest participant is 74 in our study, and the youngest are the 10-year-olds. And we've had such good feedback across all ages. The only reason for 10 is just that we wanted people who could have access to the technology and that were open to using the apps to log their symptoms. But we know children are very tech savvy these days, so I think we could definitely look at opening up to 7, 8, 9-year-olds, and we've had really good feedback, as I said, across all ages.
Any other questions? Oh, here we go.
The cause of asthma, can I'm just thinking about the quality of air in Auckland. I understand the quality of air is not very good, and the number of asthma attacks is related to that. People think we've got a nice, clean environment, but actually it's quite polluted.
Yes. Thank you for that comment. So the comment from the audience was around air pollution and the air quality in Auckland area or New Zealand, and that often people think New Zealand air is clean, but it may not be as clean as it is. And that's absolutely right. There is data that we've got from Auckland Council which looks at PM 2.5 and PM 10, which are basically looking at the size of particulate matters. And we definitely know, like near highways and motorways, and there's a clear urban-rural difference in terms of the air pollution. And that has been shown overseas to be related to asthma attack data. We're just currently in the process of analysing that. But there is definitely the hypothesis of having worse asthma if you're living in a more polluted and urban area. But there's also the other new hypothesis around green space. So in a more urban area, there's worse pollution, but there's also less greenage, so less plants. And the less greenery there is, that seems to be also associated with both the development of asthma and worse asthma control. So, excellent comment.
Hi Amy, thank you for that beautiful presentation, it was fantastic. I'm just wondering, with the increase in vaping that's come into our society, what is that impact on your studies? Is that sort of taken in? Because I know that previously it was smoking, but that's kind of been, but the younger generation has just grasped onto vaping like nothing else.
Yes, that is one of our key baseline questionnaires. When they come to see us, we ask about their smoking and their vaping histories, and we do have a number of vapors as well. In terms of how does that affect it, their physiology, we haven't looked at vapors versus non-vapors, but that's definitely worth doing in terms of do their digital markers differ. But because our alert system is personalised to the individual, so it's basically relative to your baseline, how does your heart rate and your sleep and your step count change relative to what's normal for you, then it's applicable for whether they're vaping or not, or whether they stop or not, is the baseline will just adjust if they take on vaping or stop vaping. So then the system will have to relearn what might be normal, but what we're really focusing on is that change, so in terms of detecting abnormalities, so changes in these physiological measures. But definitely that is of concern, especially the young people coming to us who have a lot of vaping. And we did also, in a separate related study, take some breath samples from those who are vapors and non-vapors to try and understand what the differences are. So watch this space. There's a lot of data that we have to analyse and work through.
Hi. Do you also collect data about the quality of air indoors? Because a lot of the housing of low quality has mold and all other sorts of particles that would affect children, of course, at the start, but also older people. So are you looking at those markers as well?
So I believe the question was around indoor air quality.
Yeah,
yeah, so just repeating it for the online audience. So the question was around, am I looking at indoor air quality, recognising that that is also related to asthma control, particularly in New Zealand with damp housing and things like that, humidity. So we did initially, when we conceptualised the study, think about whether we could put indoor sensors in people's homes to look at humidity and dampness, but then the technology burden was too high for people to be monitoring the indoor environment. So I guess in short, we're not looking specifically at the indoor air quality, unfortunately, but we do recognise the potential of like on-body sensors to monitor that.
So the smartwatches could not do that? They could not?
No, the current smartwatches can't detect indoor air quality or outdoor air quality for that matter. But maybe that suggestion we should feed back to Garmin and Apple. But maybe watch this space.
Well, thank you, everybody. So Amy has also nearly caught us up on time as well, which is just fantastic. So Amy will be around after the presentations as well. So if you've got any more questions, we can ask Amy those then. So please, everyone, if you'd like to thank Amy for her amazing presentation. Thank you. I, for one, don't think I'll be getting a smart shirt. Really, or smart socks. Really don't need to know about that sort of stuff. So I would now like to invite Associate Professor Vanessa Selak up to introduce our next guest, who is Dr. Tim Angeli-Gordon. Thank you. Oh, you can use it, yeah.
Tēnā koutou, ko Vanessa Selak toku ingoa. He aho nuku aho, waipapa taumata rau. As Sue said, I'm Vanessa Selak and I'm delighted to be here this evening at this wonderful lecture series after having heard from Associate Professor Amy Chan and to now be hearing from another wonderful colleague, Dr. Tim Angeli-Gordon. I am delighted in particular to be introducing Tim because I've had the privilege of working with him for a number of years alongside working with Tim together on the AMRF Medical Assessing Committee. Currently, Tim is working as Senior Rangahau and Innovation Manager at Te Wānanga o Aotearoa, where he's using his considerable expertise in research to train up our next generation of researchers. And it's incredible to see the amazing work that he's doing supporting that development across the Te Wānanga o Aotearoa. And he's gone from doing research that's focused on the body to research now that's looking at an incredibly broad scape, from down to the cellular level across, right across through to environmental health. However, in terms of his own research journey, that began in the University of Michigan, and that's where he undertook training in biomedical engineering. After doing research there and completing his Master of Sciences, he came to Aotearoa, and that's when he began to undertake research in gastrointestinal health, when he joined the Auckland Bioengineering Institute, which is here with us at the University of Auckland, and that was when he started to do his PhD, and that was in 2009. At the University of Auckland, his research program has focused on developing new devices and techniques that can be used to diagnose and treat gastrointestinal disorders. So I guess we've got a bit of a recurring theme this evening of the use of digital technology, except we're moving from the respiratory system to the gastrointestinal system. So thankfully what Tim has been able to do is, in addition to undertaking his senior role at Te Wānanga, is to continue his research, supported by many funders, including the Auckland Medical Research Foundation. So we're incredibly privileged to have been able to have the support to enable Tim and his team to undertake their groundbreaking, world-leading research in gastrointestinal diseases, which he'll be telling you more about. And look, before I hand over to him, I just wanted to say how wonderful it is to have Tim here. He's an incredibly humble person, but don't be fooled. He's incredibly intelligent and wise. And, alongside this commitment is an incredible strength and drive to support Maturanga Māori and Te Reo Māori and our next generation of researchers. So please join me in welcoming Tim to give his talk. Thank you.
Test. Oh, yep, thank you. Oh, well, thank you, Vanessa. It's lovely working with Vanessa, and I'm very honored by that introduction. It's always nice and sometimes awkward to be introduced like that. But I feel very fortunate to be with all of you here tonight. Tēnā koutou katoa, no mai haere mai ki te kōpapa nei. Welcome, everybody. Thank you for sticking it out. We've heard about asthma. Now we're going to move, I guess, down the body into the gastrointestinal tract, slightly adjacent, depending on where we're talking about. But typically I make a joke about the gastrointestinal tract or research being taboo, but we've already covered sweaty socks and sweaty shirts, so I'm actually feeling pretty good about my field of research at the moment. So tonight I'm going to talk to you, like Vanessa introduced, about my research in the gastrointestinal tract, particularly coming from my background as a biomedical engineer. Looking at developing new technology and devices from an engineering perspective and trying to translate those through to clinical bedside. And we'll talk about a couple examples at various stages of that process. And yeah, like Vanessa said as well, I've been very fortunate to have a relationship with the Auckland Medical Research Foundation for a number of years now, being supported very early from a fellowship and working for many years on the Medical Assessment Committee. So it's a real honor to be able to be here tonight and to speak with you all. So first, ko waio, who am I? No tia mana ahau, I'm from Germany. Engari whānau mai ahau me tupuaki ahau ki America, so I was born and raised in America. Ke Waitakere ahau e noho ana I naenei. I live in Waitakere in West Auckland now, in Henderson. Ko Bella Pullum, rawa ko manawa ora, Angeli-Gordon, aku te mahine. I have two daughters. Their names are Bella and Manawa ora. Ko Joni Angeli-Gordon, taku hoa rangatira, taku wahine. My wife is Joni Angeli-Gordon, who you can see there. And I'm very fortunate to get to actually work with her now in my current role. She is also a researcher She looks at Māori mindfulness, Māori education, and child well-being. And we're doing some quite exciting work now, bringing together my background in biomedical engineering and Western medicine with aspects of Mātauranga Māori to look at gut health from different points of view. So it's quite fun, and I think Vanessa touched on it, but this is a lot of my why. So I'd like to introduce that. And particularly, I think her, which is why none of them are here tonight, because Mono Ora, a 2.5-year-old toddler, it's about witching hour right now. I can guarantee she's running laps around the lounge now with various levels of clothing on. So I don't think you guys needed to see that tonight. And then I guess moving on to my work and who I am in my work. And Vanessa, again, thank you to her for introducing this. So for many years, I did my PhD at the Auckland Bioengineering Institute here at the University of Auckland, and then worked here for a decade building a research program in gastrointestinal health research. And part of that was founding and leading the Target Lab, so a laboratory for translational research in gastroenterology and emerging technologies, which I still lead and run today. But more recently, about 1.5 to 2 years ago, I moved over to Te Wānanga Aotearoa in the Te Manawa Haukura Research Center, which is where I work now. I'm still very fortunate. I work and collaborate closely with the university and many of those partnerships. So I guess it's a little bit of making my own path and figuring out how I can best serve a number of different communities and people. And I think that brings me to the motivation for a lot of this work. There's a very well-known whakatoki Māori or proverb that says, he aha te mea nui o te ao, he tangata, he tangata, he tangata. So what is the most important thing in the world? It's the people, it's the people, it's the people. And that's why I think for me, I like to start my talk about who I am, and really that's more about my fauna and who I'm surrounded by, who I get to work with. But in this case, this is somebody who's very special. So this is Ruby Hill. She is somebody who suffered very severely from a disorder called gastroparesis. So we're going to move more into some of the gastrointestinal health now. And gastroparesis, it literally translates to stomach paralysis. It's a very severe digestive disorder where patients suffer from abdominal pain, bloating, early satiety, which means when they eat a meal, they feel full very early, abnormally early, vomiting, and nausea. So very severe symptoms. And also in this disorder, gastroparesis, the stomach fails to empty properly. So it doesn't empty food like it should. Now, I think many people say a picture is worth 1,000 words, and I also think a face or putting an actual person to many of these disorders can be really powerful. So this is Ruby, and we'll fast forward to, this is also Ruby, not too long after. This is her in the hospital. She was diagnosed with gastroparesis after a very long and painful process with many, many ups and downs. And in the caption there, so these are all photos from online, they're publicly available, but in the caption it says Ruby at her lowest weight of 42 kgs, and that was at the time. So I think very powerful there. And then moving on, you can see, I think, probably a more powerful image. And I'll let those images speak for themselves, I think. Unfortunately, Ruby passed away a number of years ago. But I think this is particularly powerful because we actually have Ruby's mom in the audience tonight, Jo Hill, who's here with us tonight. So Jo and I met a number of years ago, and we've been working closely together in terms of just a shared enthusiasm for raising awareness for gastroparesis and working on new diagnosis and treatment options. And so I think to highlight here, this was a very difficult journey. I think you can see that in the photos. And it really highlights the challenges of the diagnosis of this disorder and also the fact that there are limited therapies available. And I think not only is that challenging for Ruby, for patients like her, but it's also challenging for clinicians. I work with a lot of medical doctors and clinicians, and those people really want to be able to treat their patient. They want to be able to give a definitive diagnosis the moment somebody walks in the door. They want to be able to give a quality treatment to make those people better. And oftentimes, the tools don't exist for them to be able to do that in a quick and efficient manner, at least not as quick and efficient as they would like, as the patient would like. So I think in many ways, we're all on the same team here. Everybody wants to deliver good for these patients, and one of the challenges is a limit of options. Oftentimes that can be technically limiting. We saw from Amy's talk, there's a lot of technical innovations that are advancing asthma, and from my perspective, it's similar in the gastrointestinal space. Now looking a little bit beyond just Ruby and that individual patient level, so gastroparesis alone, as just that one disorder, it actually affects slightly less than 1% of the global population. That's still quite a high incidence rate. If you think of 100 people that you know, one of those will have gastroparesis. That's quite high. However, it's very closely linked with diabetes, and we know that diabetes is a global epidemic at the moment. It affects about 14% of people worldwide. It's growing very rapidly, so gastroparesis is similarly growing, and it's often actually underrepresented. It's 44% more common in those diabetic individuals, so again, similar with that epidemic, it's growing along with that. And then if we look at not just gastroparesis on its own, but gastrointestinal disorders more broadly, of which many of those are applicable to what I study, they affect up to 40% of the global population. Now we're talking 2 in 5 people. If we look around this room, how many people are in here? That means we've got a handful or a couple of handfuls of people who likely have gastrointestinal disorders or symptoms or challenges. And then looking from just an economic point of view, it's hundreds of billions of dollars, looking at stats from the US alone. So worldwide, this is a really major issue. There's major resources that are contributing to this. There are many, many people who are affected. And I think on the personal level, and that's one thing I like to connect to, is looking back to Ruby, that's what those individuals look like, and that's who those individuals are. So hopefully I've convinced you that this is something that we should care about, that I care about, that I think is important to study. So looking in gastrointestinal health, so the more specific field that I study in particular is the electrophysiology, or the electrical control of the stomach and intestines. And what we're going to see here is a view from inside the stomach. So this is from an endoscope, or a camera that's been placed down the throat inside the stomach. And we can see here ring contractions that are propagating down the stomach. The stomach contracts in a ring. That ring moves down the stomach to the distal end and terminates at that end. So these contractions are what's responsible for moving food in your stomach, for breaking down food, and for moving it between organs into your intestine. Similar contractions occur in your intestine and move those down. Very similar to the heart. I think it's always nice to draw parallels in health and in medicine. So very similar to the heart, these contractions are triggered and coordinated by electrical waves that sweep through the muscle. So with your heartbeat, the actual contraction of your heart is triggered by electricity that causes your heart muscles to contract. We see that quite often on, say, medical shows on TV. The first thing that happens, you get a couple of electrodes on and you see that beep, beep, beep. That's recording the actual electrical rhythms of your heart. And we can do similar in the stomach, but it's much less developed than in the heart. So within the stomach, there's specific cell types called the interstitial cells of Cajal that are responsible for generating and propagating these electrical rhythms that coordinate the stomach. And then those are closely coupled with the muscle cells as well. And these are some images of the interstitial cells of Cajal in red, distributed within the muscle tissue, and traces showing that the interstitial cells of Cajal are the drivers of that activity, and they then drive the smooth muscle cells, or the muscle follows that activity. So I like to just sort of dig down to that detail, and I promise that's about as detailed as I'll get in this talk, because I think that's nice to know what we're looking at. We're looking at manifestations of that cellular level within the stomach. So if we look a little bit more historically, and it was lovely seeing Amy's talk, because there's many parallels, I think, and I promise we didn't talk to each other ahead of time or plan this. But I always like that quote as well, that we're standing on the shoulder of giants. I think it's lovely to look at where we've come from historically, and I think science and medical history is really interesting. So this electrical activity in the stomach is attributed to, the discovery of it is attributed to Professor Walter Alvarez. He's a fascinating man. He's a professor and also a clinician based in California in the USA. And we can see here a figure from one of his very early papers in 1922, where he's showing for the first time the electrical activity in the stomach. So there are a few things missing. We don't know the amplitude or the time duration of those signals, but what we see, those little spikes in A, are the electrical rhythms that he's recorded from the stomach. And you can see a little sketch in that lower left-hand corner of a diagram of the stomach with those lines representing the electrodes that he's placed on the stomach. He's placed some high on the stomach, and that's the top trace, where we see very little or no electrical activity, and he's placed some lower on the stomach, and that's where we see those very nice peaks that are showing the electrical rhythms that he was able to record. I love this data because I'm an engineer, I'm a bit of a nerd, I'm a bit of a tech nerd, and that is still to this day, 100 years on, one of the best electrical signals or traces that I've seen. So using what he did, it's really, really impressive. But now I get to use the magic of time travel, and we fast forward almost 100 years to the early 2000s, which is where I was doing my PhD and joined the research group that developed this technology. And we can fast forward from those couple of electrodes placed in the stomach, like Alvarez did, to now placing hundreds of electrodes on the stomach simultaneously, which allows us to look at the actual pattern of this electrical activation. Again, similar to what they're able to do in the heart, we are now moving and pushing into in the stomach at this stage. So on the left-hand side, you can see some of our electrode arrays. So there's 256 electrodes in that patch. It's 6 centimeters by 6 centimeters, so it's a relatively small patch. So it's a very tight grid of electrodes. There's 4 millimeters of spacing between those. So each of those dots is an electrode. We're able to record signals from each of those. And there's a couple of important design characteristics. And again, this was all my colleagues who came up with this. But one, those are flexible, which means they're able to adhere to the curvature of the gastrointestinal tract. And we saw in that video, it's very flexible, large contractions. So these electrodes are actually able to conform to that. And they're also sterilizable. So they're effectively plastic with electrodes, ribbons in. So the fact that these are sterilizable allows them to be translated into human patients in an operating theater, which we can see in the middle. This is in an operating theater. This is actually an Auckland City Hospital across the road. And you can see those sort of tan cables in the middle, and those are going to electrodes that have been placed via surgery onto the stomach. On the right-hand side, you can see a mathematical model or a computational model of what we saw in terms of the recordings. We're able to use these electrodes to look at the actual spatial distribution of the electrical activity in the stomach. And we can see that that actually looks quite similar to those ring contractions that we saw earlier in the video. So in the stomach, we have a single dominant pacemaker region, or where the activity originates. It's on the right-hand side, or called the greater curvature of the stomach. We get the initiation there. It rapidly forms a ring of activation that moves down the stomach and terminates at the end of the stomach. So those are all data that we were able to generate using these electrodes placed in patients. And again, this is another way to look at those data. So we can see the electrical activity moving down the stomach in those wave fronts in a healthy individual. Now, what we did from here is we also looked at diseased cases, patients who had very severe stomach disorders, like Ruby Hill with gastroparesis. And what we found was that there was highly abnormal electrical activation in those patients. So on the right-hand side, you can see a recording from one of those patients. This is a patient with gastroparesis, actually. And what we see there is we don't see those coordinated rings of activation moving down the stomach. We see quite chaotic activation. It's not originating from that single source where it normally does. It's propagating very rapidly in the opposite direction, effectively propagating up the stomach. And so we found that these abnormal rhythms, or they're called dysrhythmias, we call them, these abnormal propagation patterns were present in almost all of our gastroparetic patients, and also another disorder called chronic nausea and vomiting, which is exactly the same as gastroparesis, except the stomach empties normally. So what we found is that these electrical rhythms are quite prevalent in these patients. And that gave us, one, an opportunity to look at, can we detect these as a diagnostic to offer an early and more efficient way to diagnose these patients? And then secondly, can we correct them as a treatment option? But there were a couple of challenges with this approach. So one, it's surgically invasive, which I showed you previously. That substantially limits the applicability. Obviously, most people don't want to go get cut open to try and diagnose their stomach disorders. And then secondly, there's no current therapies for this type of activity, so that's something that we needed to develop on our own. And so just in terms of the dates, another small in the publications that we published, but that's 2012 and 2015, so it's about a decade ago, a little over a decade ago. And so those sort of challenges are what have informed the last decade of research. And so that's what I'll move into now throughout the rest of the presentation, is looking at different gastrointestinal diagnostics and therapies, specifically targeting this electrical control of the stomach. And Amy counted down in her talk. Again, many parallels, although I'm going to count up. And this isn't an actual number. So first we're going to look at minimally invasive methods for monitoring and recording this electrical activity of the stomach, so moving away from surgical invasiveness. Secondly, we'll look at gastric ablation as a possible therapy, and don't worry, I'll let you know what all of those terms mean. Thirdly, we'll look at new applications of electrical mapping of the stomach. Where are we moving in the future, and what are some new avenues that we're looking at? And then lastly, looking at precision medicine to inform personalized therapy. And again, that draws nice parallels with Amy's talk around the use of new technology and new computational aspects. So first off, looking at a new potential diagnostic in terms of endoscopic mapping. So the main research question here, if we really boil it down to a question, is can we map the electrical activation of the stomach endoscopically? So that means going down the throat, minimally invasive, no cutting, into the inside of the stomach. And so we started off by designing a device. We're able to borrow a lot of this technology and techniques from our friends in the cardiac field. Like I said, there are many parallels with electrical activity in the heart. And so we're able to use a catheter that they actually use in the heart. And you can see it here. It's got 8 different splines and 8 electrodes on each of those splines. So 64 electrodes in a sphere. What we're able to do in the stomach is add a balloon. You're not able to do that in the heart. The heart doesn't really like when it's not able to fill with blood, but we're okay to do that in the stomach. So we add a balloon to the inside of that catheter. That allows us to get very good contact with the inside of the stomach. And then that whole catheter, in the bottom left, you can see it actually pulls down and narrows right down. So we're able to drag that down the throat with an endoscope, place it inside the stomach, inflate it, which we can see on the right-hand side from one of our early trials in a patient at Auckland City Hospital, again, right across the road. So that was our device. We started off doing preclinical validation. Again, it's very important to validate this stuff before we go putting it in patients. And we're able to show that we can record activity from inside the stomach on the bottom side, the mucosa, that's the inside of your stomach, and that compared very highly or very closely with our normal surgical electrodes placed on the outside of the stomach or the serosa. So we're able to validate that we're able to use this device to record data, that the data is accurate based on our surgically invasive gold standard, if you will, measurement techniques. But one of the challenges was we were generating a lot of data, and the data was quite a different structure than what we're used to. So that was a little bit of a step back now to the engineering side, and we developed custom software. This is a PhD student, Alexander Chan, who, as his PhD, developed an entire custom software program and platform to be able to analyze the data from this new device, the unique spherical geometry, the different electrode orientation, and to allow us to actually analyze those data. What we could see then is that that program allows us to create these maps, again, showing the movement of this electrical activity, in this case from red to yellow, moving down the stomach, and to then push this device into larger scale trials. In this case, it was moving into a first-in-human trial in clinic. And this is a photo of us. This is the first ever patient that we put this device into, again, across the road at Auckland City Hospital. That's me on the right, a younger version of me. And then we've got a gastroenterologist on the left, some great nursing staff, and a patient lying on the table. And you can see on the monitor in the back middle of that photo our devices in the inside of that patient's stomach. So that was quite an exciting day. I'm very thankful to Greg O'Grady, who's a close colleague and friend, who was in the room and took that photo, because I didn't think of it, and he had the presence of mind to think, wow, this is a really fantastic step. It's one of my favorite photos because, to me, what I've always dreamed about in biomedical engineering is developing something new and putting it into clinic, and this was the day that actually happened. So it was really exciting to do that. We then moved that on, so we did a feasibility trial of 13 patients, like I said, at Auckland City Hospital. David Robotham is a gastroenterologist that we worked very closely with. He's absolutely fantastic to work with. And so we completed that trial and published it last year. We're able to record electrical signals, and in this case we can see where we're able to see where that activity actually originates at that star. So this is looking at the actual electricity at each of those electrodes. The red dots are where the actual event is, where the wave meets the electrode. And then we're able to create these maps. So again, this shows the electrical activation from red to yellow. And again, marked with that yellow star, that's actually a site of activation that in this case is low in the stomach, which is an abnormal activation pattern, like I showed earlier with our surgical mapping. So what this shows is that even in these very early trials, we're able to actually detect these abnormalities with this minimally invasive technique, which was a really exciting outcome. So can we map the electrical activity endoscopically? Yes. So the next steps, can we correlate that activity with symptoms? And this becomes a really important question clinically, because that's how we can push then towards a diagnosis. Long story short, we're not quite there, but we have secured most of the data. We're looking at finishing that and analyzing those data at the moment. In that case, we've moved or expanded to a multi-center trial at both Auckland and Christchurch, working with a second gastroenterologist, Gary Lim. Again, really fantastic. And we're working on those, finalizing those data in the manuscript now. But it looks like we can start to draw some nice correlation between symptoms and electrical activity. So that was a great outcome. We moved towards a minimally invasive option for diagnosis. So moving on to our next sort of point is, can we use this electrical activity as a therapy or as a treatment? And in particular, looking at gastric ablation as an option for that. So if we look at ablation, again, borrowing from my friends in cardiology, if anyone's here who works in the heart, thank you. So ablation is a technique that's very, very commonly used in cardiac therapy. Effectively, ablation is delivering energy, focused energy to the tissue to effectively burn the tissue or damage the tissue in a way that creates a scar that's not able to activate electrically, and that can be targeted at those abnormal sites to eliminate the abnormality and allow the normal propagation to restore. In the heart, it's done by running a catheter up typically through your femoral artery in your groin, up into your heart, targeting that to abnormal activation sites and eliminating those. We effectively borrowed all of these techniques from our friends in cardiology and moved them into the stomach, which hadn't been done before, despite many of the parallels. So again, boiling it down to a single question, if we could, can we use ablation to eliminate abnormal electrical activation in the stomach is what we were looking at. So this is another PhD student, Zara. She had a fantastic PhD, and she tackled this question or this problem for her PhD. So we did this by initially validating the set of parameters or energy doses that were effective for blocking the electrical activity in the stomach. We then set out to target sites of abnormal activation. We initially mapped the activity of the stomach using the techniques I showed earlier. We're able to identify abnormal sites in a subset of, in this case, it's a preclinical model. We use pigs in that case because they have very similar stomachs. So we're able to target sites of abnormality. We're able to mark those sites, then target them with ablation. So in this case, we performed a box ablation around that site. We then mapped after the fact, and you can see an image from us doing that. So again, this is open surgical case. And what we're able to show, I realize this is quite complicated, but effectively on the top, if we look at that nice rainbow map, The activation is in the red region. That's an abnormal activation, low on the stomach compared to where it would normally be. In the bottom, we've got that nice black box, which represents our ablation, and we can see that the red activity is coming from the top to the bottom in the stomach, effectively showing that we've eliminated that pacemaker region, that abnormal region, and we've restored the normal propagation, similar to those animations that I showed you early on. We published these data, and I always like to show that journal cover on the right, because that's actually, Zara's a fantastic artist. That's actually, I think, her true passion. She sort of fell into bioengineering as something to do when she wasn't doing her art. And so she actually designed, that's artwork that she designed. It's a tessellated image of stomach tissue that she then colored based on different types within the tissue. And we submitted it to the journal, and they ended up running it as a journal cover as a bit of a sort of metaphor for the connection between the gut and the brain as well, because it looks quite similar to brain images. So I always love, I talked about the personal connections and the motivation and people. I think it's really lovely seeing, especially students, bring their different passions into the work. So that's just something that's fun that we sort of did along the way. So can we use ablation to eliminate abnormal electrical activation in the stomach? Yes, we can, which again was very exciting. But one of the challenges here is those were all done in single-day studies. So if you talk to especially clinicians in the gastrointestinal tract, they get very nervous when you're putting heat and you're burning the gastrointestinal tract. That makes them very uncomfortable, which I learned very early on. And rightfully so, because there can be some really catastrophic effects. And actually, one of the major complications of cardiac ablation is if you get too deep of a burn that goes through to the esophagus, you can get issues with your esophagus afterwards. So what we wanted to then look at, and this is really, really critical if we look at the translation of this technology, is does it remain safe and effective in the post-surgical recovering period? So we set up a series of studies where we extended them out to 2 weeks post-surgery. That time frame is quite important because typically if you have a post-surgical complication in your stomach and you have a perforation, that's the most severe, where effectively you have a scar that rips open. That typically happens within 4 to 5 days post-surgery. So we're able to cover that period and then extend out to 2 weeks, which is also when surgically we see a reconnection of those electrical networks. So if we cut the stomach and rejoin them together, they reconnect after 2 weeks. So we're also interested, not only do we have safety in terms of a lack of perforation, but do we get a reconnectivity of that electrical activity, because that would not be good for us. We want to eliminate that activity long term. So we did a very similar study. We performed an ablation. We recovered for 2 weeks. And we showed that in these cases, we still had that conduction block present after the 2-week period. We didn't have any adverse complications during the post-surgical recovery. No perforation, no signs of progressing towards perforation. We were well on the way towards the healing process. So again, that was exciting because that shows that this is potentially translatable longer term. But again, and not to be a bit of a downer, but there is a challenge here in that it's invasive surgery. And so that sort of motivates our next step. But first, does it remain safe and effective? Yes. I always like to celebrate these wins. So then, as I said, it's still surgically invasive. So the next question was, can we perform this ablation endoscopically? So talking about our mapping endoscopically, can we also ablate endoscopically? That'd be pretty cool, right? So again, got a great PhD student on board, Ashton. He tackled this problem for his PhD. Recently defended his PhD, and he's graduating in a couple of months. Had a fantastic outcome, so that was really exciting as well. So looking at what we did here, we used our gold standard invasive electrodes. So we actually did perform surgery on these animals to record the electrical rhythms from that serosal surface, that surgical side. But we performed all of the ablation endoscopically. So we placed the ablation catheter down the throat into the stomach endoscopically, minimally invasively. We performed ablation, and then we also collected the tissue to look at what that ablation did and the damage done. So we can see an image on the left. That's the ablation catheter on the inside of the stomach from the endoscope. Performing our ablation, that little bleached white tissue is the actual burning of the tissue effectively. And then we can see some results on the right-hand side. So these are a little bit challenging to talk through, but again, we start at the dark red. We see in the baseline that we have propagation from the top to the bottom of the stomach. Now in this case, we actually induced an ectopic pacemaker or induced an abnormal pacemaker with pacing so that we could ensure that that was present and ensure that it was in the same location. We then targeted that location with ablation, ablated in that location, and on the right-hand side, you can see we've got our ablations in the middle, and we've restored that activity, dark red at the top, moving down the stomach to the normal pattern. So we're really excited about this because we've now translated not only our mapping to go endoscopically, minimally invasively, but also our ablation technology. And we're carrying this work on as part of a new grant funded by the Health Research Council. So again, very excited with that work. So can we do it? Yes, we can. You might see a theme here. It's nice to celebrate the wins occasionally. So moving on, and we're getting close to wrapping up, so I think we're on time. But looking now at sort of our third aim, our third talking point for today is looking at new applications. One of those being the pylorus. So the pylorus is also called the pyloric sphincter. It's the barrier between your stomach and your intestine. Now it's very important because that's how your stomach empties. It controls your stomach emptying. There are a whole range of disorders where the pylorus malfunctions. And we're interested in understanding, can we develop or apply some of our technology to those specific disorders? One of those cases is in post-surgical complications. So a couple of my colleagues there, I mentioned Greg, he was the one who took that lovely photo of me in the endoscopy theater, and Tim Wang, who was a PhD student as well, and did this as part of his PhD. So we had quite a unique opportunity where a patient presented to clinic who had previously had a surgery in their stomach, and I won't get bogged down in the details, but effectively they had the bottom of the stomach cut out and the top of the intestine. They were then rejoined. So what that does is it actually removes the pyloric sphincter, and you get a reconnectivity of the stomach to the intestine. The reason this is important, especially from our sort of nerdy little focus in the electrical activity, is that not only does the pylorus act as a barrier, a mechanical barrier of food and contents, but it's also an electrical barrier. So the electrical rhythms from the stomach don't propagate through the pylorus, and the electrical rhythms from the intestine don't move up. But when you remove that, we were interested in whether, potentially, some of the symptoms for this patient were caused by abnormal propagation, or whether there was abnormal propagation in the patient. We can see these are images of the reconnectivity from A being the stomach remnant, B the scar tissue, and C being the intestinal remnant. And it's quite interesting. It always looks the opposite way around because the intestine is typically quite narrow and the stomach is typically quite large, but we actually saw an enlarging of the intestine as part of the outcome of this. So it's just, again, a little nerdy aside. But then, again, we mapped the electrical activity in this patient, and we did see that red line indicates the scar tissue. This is a scar that's 20 years old, so it's a very long-standing surgery. We saw activation in this patient coming from that intestinal remnant through the scar, moving up in the opposite direction, upwards the stomach, or retrograde up the stomach, and then that carried up higher in the stomach remnant, continued in the retrograde direction. So we saw consistent abnormal activation in this patient caused by the removal of that pylorus. And we sort of framed that as a gastrointestinal aberrant pathway, or this post-surgical complications that can create abnormalities. So that was an interesting finding and discovery on its own right. And I think if we look back to the ablation technology, there's an opportunity there to actually try and reintroduce that conduction barrier at that site, potentially as something to look at in the future. But we're also quite interested in looking at the pylorus in more detail. So we developed some custom electrodes specifically informed by the anatomy of that area, because that's one of the technical challenges of recording. And we're able to record in quite high detail the electrical activation of not only the intestine and that proximal intestinal pacemaker, the electrical conduction gap at the pylorus, and then also the activation at the stomach. We also correlated that with mechanical recordings placed inside the lumen of the intestine. And again, this was a fantastic PhD produced by Sam. So looking at trying to understand more about function of the GI tract using some of these techniques. Which brings me to our last talking point, being precision medicine for personalized care. So one of the challenges in medicine is that we often have a one-size-fits-all approach or a one-size-fits-many approach. But every patient is different. They all appear different. And I think particularly in the gastrointestinal field where we have relatively few symptoms to diagnose, and we have very few biomarkers that are known, so very few things that we can actually measure from these patients in comparison to some other medical fields, we end up having relatively heterogeneous patient populations. So on the left-hand side, we can see we can separate patients out into, say, irritable bowel syndrome and functional constipation. But what we actually have in effect is we've got a whole range of patients that have different disorders or different physiology within those. So our question is, can we use technology, in this case AI, artificial intelligence, it's sort of a catch-all. We hear a lot about that these days, some good, some not so good. But what we wanted to do was use very specific computational techniques to see if we could try and separate out with more accuracy these different patient groups. And we're able to do that using a pretty unique data set with some colleagues in the National Science Challenge on High-Value Nutrition. And again, that was work led by a fantastic collaborator that I work with, Jared Dowrick, who's leading a lot of that. And then also looking at different ways of applying some of our engineering techniques, and in this case, using computational models. So can we actually, on the left-hand side, I realize it's a very complicated figure, which Jared apologized about as well, but I think that the take-home point is that we're able to actually represent many, many different processes of the body. Each of those little squares in that figure is a different process that we're able to represent mathematically. We're able to then link those mathematical representation of the different processes to build effectively a system or a physiological system within the body. So the end goal is to create these digital twins which represent the human body and allow us to test digitally or computationally these different interventions. That's important to me. I'd like to do less work, say, with animals in the preclinical setting. Very important to do that work, but we always like to minimize that and to get the maximum efficiency from our results. So this allows us to then input some of our new discoveries into these computational methods, build these digital patients to try and improve the precision of our healthcare. And again, that's great work with Jared Dowrick. So I think just to wrap up and I guess re-summarize what we've talked about, we've looked at new diagnostic options in terms of going endoscopically down the throat to detect these abnormal electrical rhythms and these very severe gastrointestinal disorders. We've looked at developing a new therapy that can actually eliminate the abnormalities that we've seen in these patients, and we've pushed that towards a minimally invasive endoscopic approach as well. We've looked at new applications, particularly around the pylorus and the role of the pylorus, and where we can move towards that in the future. And we've looked at applying a lot of the different engineering principles into improving precision health. And I think those are each represented and were presented as independent pillars, but I think, as I touched on earlier, we can see many parallels here in terms of the endoscopic diagnosis, feeding into the endoscopic treatment, the fact that we found the pylorus could actually benefit in some of those post-surgical cases from some of the treatment we developed, and then we're able to actually amplify the impact of this using the precision health. So we can see that this is actually not independent silos of research, but a whole program that we're really excited to develop. And I think it all centers on, again, those patients. So linking back to Ruby and the team that worked with her and looking at that. Can we help Rubies? And I think the answer is yes, which is exciting for me. So thank you to many of the collaborators I work with. Some of those you'll know if you work in the university, some international collaborators, and this is really just a few of the highlights from some of those key studies that I talked about. A lot of different expertise there. So engineers, a lot of engineers on the left, some great technicians as well. So Lindley, I always like to highlight her. She's a technician that we work with. She's fantastic. We always joke that she could probably do all of our studies better than we can do them because she's watched for decades us do that work. Some great clinicians based in New Zealand and some fantastic academic clinicians overseas that we're very fortunate to work with as well. And then lastly, just a thank you as well to all of the funders. I think particularly tonight, that top line, Joe Hill from the Ruby's Voice Trust has been a supporter of our work recently, and absolutely the Auckland Medical Research Foundation tonight, and in parallel, the Kelleher Charitable Trust, who funded some of this work. And I think this is the last slide, so I think it's, again, an honor to be able to present tonight. But I dug this up, so this was an email. If you can see the date, it's Tuesday, 8th of November, 2016, at 11 a.m. I first thought it said 11 p.m., and I thought Hannah was working late that night, but 11 a.m., and it says your AMRF application has been approved. And so this application was for my first postdoctoral fellowship, or my first independent fellowship, which was just under $200,000, and that funded the very initial foundation of the endoscopic electrical mapping. So I think it's, I'm always very thankful to the AMRF. I always remember this because this is effectively the funding that launched my career. And I hope it was an exciting journey to sort of look at what that 2 years and that funding amount ended up building into over the next decade beyond that. And like I said as well, I was very fortunate to serve on the medical committee for many years. I stepped down earlier this year, mainly due to that 2.5-year-old that I've got running around at home. But they all know that I'm only a phone call away. So yeah, thank you all for coming tonight, and I'm happy to take any questions.
Thank you, Tim. Another amazing presentation, and I think behind Tim's work, for as many yeses as you saw up there tonight, there was probably thousands of no's as well. And I think Tim's passion and his drive comes out as to what happens to make a yes occur. And behind that is a person like Ruby, both for Amy and for Tim. They are working tirelessly for people, for our health, for our better health. So we can't thank either of them any more than we can. So thank you again.
Sue.
Time for questions. Any questions? Oh no, not that. Hello. Oh. I was going to throw this up to Frank, but no. Thank you.
Hi, Tim. Thanks. Great talk. I was just wondering whether you're developing a pacemaker for the stomach.
Oh, yeah, fantastic question. So just for the audience online, the question was whether we're developing a pacemaker for the stomach. The short answer is yes. I think that's another sort of parallel technology that we can borrow from our friends in the cardiac field of actually trying to drive the electrical activation of the stomach or pace the electrical activation of the stomach. So it is, we've been working for, again, about a decade on that. We've developed some custom pacemakers for the stomach. Some worked on different settings that are effective for driving that activity. We have done some initial trials in human patients, and then that's been translated as well to the intestine. And I didn't really touch at all on the electrical activity of the intestine. That's actually where I did my PhD, was in the intestine. And then I moved up to greener pastures in the stomach. But looking at, there's some good work going on with a recent PhD graduate looking at intestinal pacing as well. So that's done by many of my colleagues. Leo Cheng, Professor Leo Cheng at ABI is leading that. But yeah, great question. It is an area that we're looking at.
Question up here. You have to hold that up. Right.
I just wonder how many abnormalities somebody might have in their stomach. Maybe there's probably more than one, wouldn't there be?
Yeah, great question. Again, for those online, the question was around how many abnormalities someone may have in their stomach. So it's a fantastic question. And we have looked a lot at that, at actually classifying the different types of abnormalities. There are many different types of abnormalities. Historically, if we look back, it was largely looked at the frequency of activity. So how many actual electrical waves per minute are moving in the stomach. And that was, again, largely because of the technical limitations. So if you're only able to record from one or a couple of locations on the stomach, that's all you can really look at is the frequency of that activity. So again, similar to cardiology, they looked at elevated stomach rates and lower stomach rates, and there were patients that were in either of those. What we found in our work, and one of the really important factors, is that actually in many of those patients with normal frequency pattern, we found abnormalities in the actual spatial propagation. So like I showed, there are abnormalities where there's activation low in the stomach and that's propagating up the stomach. That's an example that many of those patients have the normal frequency, but it's very, very different to the normal pattern. So that was one of the benefits of this higher resolution approach. Looking at actual incidence rates, that retrograde propagation, it looks to occur in about 30 to 35% of patients. So it's quite a substantial cohort that have that particular pattern. But that's, again, an ongoing area, is looking at these individual patterns. What can we detect? What can we diagnose? And what does that mean in terms of patient numbers? Yeah, great question. Thank you.
Where's Jess? Oh, might be easier to go.
Come in, Christine.
Thanks, Tim, for your good, clear presentation. Learned a lot. Just wondering, ethnically, are there certain ethnic groups more predisposed to stomach disorders than others?
Oh, yeah, that's a great question as well. So the question was, ethnically, are there certain ethnic groups that are more predisposed to higher rates or higher incidence of stomach disorders or gastrointestinal disorders? Very good question. I don't know too much about that. I do know that there are higher incidence rates in Māori, in Pacifica. Those are groups that we look at a lot in New Zealand, and there are higher incidence rates in those groups. I'm not too sure about other ethnic groups. I do know that, interestingly, there are much higher incidence rates in females compared to males. And particularly if we look at gastroparesis, that's actually a disorder that affects females at a much, much higher rate. I think it's actually about 3 or 4 to 1 compared to males. And also affects young females as well. And I think Ruby's an example of that, of how that disorder is affecting that certain population. So I think, again, it's an area of interest looking at those different demographics. Something that we're not really looking at the incidence rates, but I did mention that we've partnered with a number of Māori colleagues, and I use colleague in a very loose term because my wife is one of those that I get to work very closely with now. But looking at not necessarily the incidence rates, but what can we learn from Māori culture and Māori knowledge in this area. And one of the interesting things we've found is that we talk about the gut-brain axis. So a lot of what we study is this connection between the gut and the brain and how those electrical signals are integrated. This gut-brain axis it's a real hot topic in the field in sort of Western medicine now. But if we look at Te Ao Māori and Te Reo Māori in particular, we can see that there's a real connection with the gut and the brain. So an example of that is the word for the stomach in Māori is puku. The word puku is also a root word or an accentuating word for many emotional terms or neurological type terms. So pukuriri means furious. So there's this sort of inherent connection in the Māori language between the gut and the brain. So we're starting to look at that and how that looks from that Te Ao Māori approach, and how we may be able to use some of that Mātauranga Māori as a treatment or as an intervention as well. And I say we as well because my wife's leading that work, so yeah.
Hi, any thoughts on the reason why the activation site is upside down or in the right? What causes that to happen?
Oh, yeah, that's a very good question. So the question was, any thoughts on why the activation site is the opposite way around in some of those cases, where the activation, that electrical activation, is actually low in the stomach rather than higher in the stomach? Long story short, no. There has been a lot of work done over many decades, and even with some of the lower resolution techniques, there were people who were hypothesizing decades ago that there was activation from low in the stomach, and there was actually, some people thought, a secondary pacemaker region lower in the stomach. What we do know is that there's no difference in the cell type, so they are consistently the same cells in those locations. There is a different inherent frequency that they operate at, so there's more proximal cells, the higher up in the stomach, have a higher frequency, and that causes them to drive and entrain the cells lower down in the stomach. So effectively, that's a mechanism by which that pacemaker originates high. We know that disruptions to that network can allow activity lower in the stomach, but it's really an area of ongoing research why that occurs. Yeah. Any other questions? Thank you, everyone.
So I think both the presentations tonight have, they've said so much in the presentations and said it in such great terms, in terms of lay terms and us being able to understand it, that the range of questions just haven't had to be asked. But I would like to invite both Tim and Amy back up here just so we can say a little thank you. Both Tim and Amy have received fellowships from Auckland Medical Research Foundation over the years, but these two are the reason we exist. We exist to support particularly the career pathways of researchers, and that's the reason we can have so many yeses to these really, really complicated questions that are being asked, and the years and years of work and passion that goes into it is absolutely extraordinary. So I'd just like you all to put your hands together to thank these two extraordinary researchers. Thanks everybody online for joining us tonight, but thank you everybody too for coming in person to this third speaker series event, and thank you too for our 70th partners that are here tonight. And yeah, please keep on supporting Auckland Medical Research Foundation, because actually these are the people that we're supporting. So I wish you good night.